Hi group, today we're going to talk about the immunoassays, and these are methods used to look for antigens or antibodies, and here we can use the immunoassays to help diagnose things like measles, mumps, HIV, hepatitis B, West Nile, avian influenza, and all kinds of diseases, and we can also look for antibodies in patients' serum. Okay, so we're going to talk about two methods today. We're going to talk about ELISA, which stands for enzyme-linked immunosorbent assay. Here we have antigen and antibodies that are labeled with enzymes, and we're also going to talk about immunofluorescence, so stay tuned. There's two types of immunoassays I want to talk about right away, and that would be the direct and indirect ELISA methods, and that stands for enzyme-linked immunosorbent assay. ELISAs or EIAs are for antigen detection, such as hepatitis B surface antigen, and there's 4 steps. Antigen-specific antibodies attached to a solid phase surface, those are plastic beads, and the patient's serum that may contain antigen is added to that specimen. Next, an enzyme-labeled antibody specific to that conjugate is added. Finally, a chromogenic substrate is added, which changes color in the presence of the enzyme. A lot of times that color is yellow, and you can see that with the naked eye. The amount of color that develops is proportional to the amount of antigen in the patient's specimen. Now, we can also detect antibodies. There are 3 types of methods for antibody detection called non-competitive, competitive, and capture antibody detection. Okay, in indirect ELISA, a plastic plate is coated with antigen. This is also known as solid phase technology. An antigen reacts with the antibody in patient's serum. The plate is incubated with enzyme-labeled antibody conjugate, and if antibody is present, that conjugate reacts with the antigen-antibody complex on the plate. The enzyme activity is measured spectrophotometrically after the addition of chromogen. And again, using the spectrophotometer, we're measuring light relative to wavelength. The enzyme either absorbs or transmits light at certain wavelengths. And again, we have direct ELISA, which is where we have antibody and conjugate on cellulose acetate paper. One example of a direct ELISA test is the strep throat antigen test. And then we have indirect ELISA, where we have antigen in wells. We incubate the sample with substrate. We add peroxidase, and if antibody is present, the substrate will change color. There's 2 enzyme immunoassay approaches when we are looking for a disease, okay? We have testing for specific antigens or testing for antigen-specific antibodies. So we can look for antigens in the patient's serum or blood, or we can look for antibodies in the patient's serum, okay? ELISA, or enzyme-linked immunosorbent assay, is the method, and this is also known as EIA, or enzyme immunoassay. ELISA is designed to detect antigens or antibodies by producing an enzyme-triggered color change. This method is performed usually in a 96-well plate, and this is known as an indirect ELISA method. An indirect ELISA method is usually reported by some sort of numerical result in a calculation from the optical density reading. Enzymes used in this methodology help detect or quantitate immunologic reactions. So these enzymes are coupled with conjugates, and a conjugate is a dye, giving us a visual clue. And again, that visual clue is often yellow in nature, and it gives us an indication as the strength of the antigen or antibodies in each well. The higher the optical density, the more strongly positive the well is. And again, you can see that with the naked eye, where if the well is very strongly yellow, it means that there is more antigen or antibody in that sample. Now, the enzymes that are used, these are unlabeled antigen and unlabeled antibodies, and there's a labeled antigen, a labeled antiglobulin, or a enzyme that is used in this approach. We're going to use either horseradish peroxidase, alkaline phosphatase, glucose 6-phosphatase dehydrogenase, or beta-galactosidase. Now, anytime we're doing an ELISA in the laboratory, we have to have a known or reference sera to compare our optical density to. So if our optical density is higher than that of the control or reference sera, then that sample is probably positive for antigen or antibody. Okay? One example of antigen detection is looking for the hepatitis B surface antigen, where the antigen-specific antibody is attached to a solid phase surface like a plastic bead or well. The patient's serum may contain the antigen, is added to the bead, then an enzyme-labeled antibody specific to that antigen conjugate is added. A chromogenic substance is added which changes color in the presence of that enzyme. The amount of color is proportional to the amount of antigen in the patient's serum. So the darker the color is, the more antigen that is present in the patient's serum. And you can see that in the hepatitis B surface antigen test. Now there's three types of antibody detection that we can do with ELISA, and those are called non-competitive, competitive, and capture ELISA. Non-competitive ELISA uses excess labeled specific antibody towards the analyte of intent, while competitive ELISA uses a labeled analyte. It looks for concentration of antigen, and labeled antigen and sample antigen compete for binding of antibody. In capture ELISA, we use capture antibody which is specific to the target antigen, and this is also known as sandwich ELISA. Okay, so we've got our non-competitive, competitive, and capture ELISAs. So our non-competitive enzyme immunoassay takes place with a specific antigen is attached to a solid phase surface, such as a plastic bead or microtiter well. This patient's serum then could contain antibodies such as cytomegalovirus, IgG, or human immunodeficiency virus, is added to the solid phase surface, followed by an enzyme-labeled antibody specific to the test antibody. The added chromogen substance changes the color if the enzyme is present. Then we have competitive enzyme immunoassay. Competitive enzyme immunoassay involves using a solid phase surface to which a specific antigen is attached. If the antigen to be measured is small or only has one epitope for antibody binding, a competitive method is used in which either the antigen is labeled and competes for the unlabeled antigen-antibody complex formation, or the antibody is labeled and competes for the bound antigen and antigen in the sample. In capture enzyme immunoassay, again, this is also known as sandwich ELISA. It's designed for a specific type of antibodies such as IgM or IgG. Okay, the patient specimen potentially contains IgM or IgG antibody. Specific antigen is then added, and finally a chromogenic substance is added which changes color in the presence of the enzyme. The amount of color that develops is proportional to the amount of antigen-specific IgM or IgG in the patient's serum. When we're doing an ELISA procedure, we want to go ahead and look for an optical density that's higher than the reference sera. In antibody detection for infectious diseases, we generally first have a well that has no antigen or antibody, and that's called the blank. We must read the optical density of the blank, and then we usually run two standards and come up with the average of both those standards. We find the mean of the two standards optical density and divide by our patient sample optical density by the mean of the two standards minus the optical density of the blank. There's an example below, and that determines how you can see if the patient's wells are positive for antigen or antibody. So anytime you're doing these tests in the serological laboratory, you always want to make sure that you consult the package insert. The package insert will tell you the optical density ranges for what is considered to be negative, what is considered to be equivocal, which is the gray zone, or what is considered to be positive. Depending if capture ELISA is used to detect IgM or IgM-specific antibodies, this is going to determine if the disease is in the acute stage or the convalescent stage of the disease. Remember, IgM is usually increased in acute diseases, while IgG is usually is more positive in convalescent stages, or if you're immune, if you've had a successful vaccination. And here's a nice link that you can use to watch the ELISA procedure. ELISA is a common immunoassay used in serology laboratories to help diagnose the following infectious diseases. One would be Lyme disease, cytomegalovirus or hepatitis A or B, HIV, sugar toxins, E. coli 0157H7 and things like that, Clostridioides difficile, West Nile, measles, mumps, toxoplasmosis, and those are just a few that we can use ELISA for to detect those infectious diseases. So here's a picture of solid phase, or our 96 well plate, and you can see if we had several patients, you would put the blank in the upper left, and that would be A1, and then you would put the two calibrators together, because remember, we're going to run the calibrators and find the mean of the two calibrators. Then we have our negative control and positive control, and then our patients go in the wells following our controls. Fluorescent labeling is another method used to demonstrate the complexing of antigens and antibodies to a specific region of a cell to allow for identification, such as in flow cytometry analysis of blood cells. Fluorescent molecules, fluorophores, are used as substitutes for radioisotopes or enzyme labels. Fluorescent techniques are extremely specific and sensitive. We can use fluorochromes, which are a chemical that can absorb light energy at a short wavelength and almost instantaneously emit light at a longer wavelength. A fluorochrome that is chosen is usually one that can absorb UV or short blue light and emit light in the visible range. The fluorochrome is activated or excited by the particular wavelengths of light. The fluorochrome we usually use in immunofluorescence technology is called FITC, or fluorescein isothiocyanate. The FITC is blue and allows for maximum absorption, so emission is in the yellow-green range to which the human eye is highly sensitive. So to us, observing using the fluorescent microscope, the antigen or the substance that we're looking for is going to fluoresce apple green. Now, some rhodamines fluoresce on the red portion of the spectrum. This fluorescent compound has an affinity for proteins. There are two types or two methods of immunofluorescence. One is called direct immunofluorescent assay, and another one is called the indirect immunofluorescent assay. In the direct fluorescent antibody technique, also known as the DFA, a conjugated antibody is used to detect antigen-antibody reactions at a microscopic level. DFA can be applied to tissue sections or in smears for microorganisms. In addition, clinical immunophenotyping is done through direct fluorescent assays. In indirect immunofluorescent assays, the basis for indirect fluorescent assays is that antibodies or immunoglobulins not only react with homologous antigens, but can also act as antigens and react with anti-immunoglobulins. IFA is the serological method that can be used for the detection of diverse antibodies, primarily in research laboratories. Immunofluorescence is used extensively in the detection of autoantibodies and antibodies to tissue and cellular antigens, for example, anti-nuclear antibodies. The direct fluorescence antibody test, we have an antigen, or it's the unknown element, plus specific conjugate. It is primarily used to detect nucleic acids of organisms such as CMV, RSV, hepatitis B, gonorrhea, and chlamydia. While indirect fluorescent antibody testing, we have an antigen plus patient serum that is incubated. We add our fluorescein isothiocyanate labeled anti-human gamma globulin. That's the conjugate or specific antibody attached to a fluorochrome, and we incubate it again and observe for fluorescence or no fluorescence. Here I've got a slide that helps you compare the differences between direct fluorescent antibody and indirect fluorescent antibody. In direct fluorescent antibody, for example, if we're looking for RSV, you have a primary antibody that is connected to the antigen on a cell and a fluorophore or fluorochrome that is connected to that primary antibody. In the indirect fluorescent antibody test, we have a cell with an antigen attached to it. We have a primary antibody and secondary antibodies with fluorophores that are connected to that primary antibody. There are definitely advantages and disadvantages to fluorescent antibody testing. Some advantages is that it's a very rapid method. It's a very rapid method for identifying antibody. It may eliminate the need for culturing things like gonorrhea and chlamydia. A pure culture is not necessary. You can locate even small numbers of organisms because the procedure is very simple and very, very sensitive. Some disadvantages of fluorescent antibody testing is that it's very expensive. You need a fluorescent microscope, and you need personnel that have a high degree of expertise in fluorescent microscopy. It takes a little bit of getting used to if you haven't done fluorescent microscopy for a number of months to years. The fluorescent microscope is quite a complicated and expensive piece of equipment. It includes such parts as the condenser, the oculars, the objectives, a heat filter that's located between the light source and exciter filter, and that actually removes the heat energy from the light source and protects the other filters. We have the exciter, which is located between the heat filter and specimen, stops visible light and allows ultraviolet light to pass through. It transmits the wavelength of light compatible with the fluorochrome. The fluorochrome is going to absorb the light. We have the barrier, which is located between the specimen and the observer, and it transmits wavelengths emitted by the specimen and retards the light below 500 nanometers. And then we have the light source. Okay, and incident light is what we use in most laboratories, where we have the light, and then between the exciter filter and the light, we have the heat filter, and then we have the exciter filter. And then we have a beam splitter with built-in suppression filters and beam splitting mirrors that actually goes between the ocular and the objective above the specimen. Again, we have two types of fluorescent microscopy. We've got transmitted light and we've got incident light. In transmitted light microscopy, the light is passed through the specimen, but it's partially absorbed. Here we use a dark field condenser, and the specimen has to be viewed on oil. In incident light, the light is focused on the surface of the specimen, and there's no light that's actually absorbed. No condenser is needed, as the objectives also function as the condenser. The light beam splitter reflects the shorter wavelengths down the objective and transmits only longer wavelengths back to the ocular. And then here's a diagram of an incident light fluorescent microscope, and I'm just going to leave this slide up for just a second for you to look at. And then here's another picture of a fluorescent microscope, and it outlines the different parts of the fluorescent microscope. So I'll give you a few minutes to look at this. So most of the time in most medical serology labs, we're using ELISA to identify diseases or antibodies in patient serum, or we're using indirect or direct fluorescent antibody testing. But there are some emerging fluorescent labeling technologies. One is called quantum dots or Q dots. Here we have a semiconductive nanocrystal that is used to fluoresce biological specimens. Here, the quantum dots look for differences in crystal size production and different fluorescence signals, allowing the tech to look at many analytes with a single assay. Then we have the SQUID technology, which is superconducting quantum interference device. Here we have antibodies that are tagged with super paramagnetic particles. The amplitude of tagged particles is proportional to particles that are bound. Then we have LOCI, which is luminescent oxygen channeling immunoassay. We have two latex particles and a sensitized particle or donor bead that absorbs energy and is used in combination with a chemiluminescent molecule or a receptor bead. The oxygen moves from one bead to another bead, triggering a luminescent signal when particles are in proximity during excitation. And then we have FISH, fluorescence in situ hybridization, where we use fluorescent molecules to brightly paint genes or chromosomes. So those are the emerging fluorescent labeling technologies. So that's it. That's ELISA and immunofluorescence in a nutshell. Be sure to read the chapter on immunoassays, and this will be a little bit easier for you to understand when you have all those images in front of you in the textbook.